So we work on HIV in my group, and the therapies that are used to treat HIV are called anti-retroviral therapy. And this is because they are designed to inhibit distinct steps of the HIV life cycle. So some therapies may inhibit the virus from binding to your cell, like an entry inhibitor. Other therapies may inhibit different viral proteins, like reverse transcriptase or integrase, that are critical for the virus to replicate. These therapies are given in a combinatorial fashion. So when that's happened, they're called CART. And when this occurs, these therapies have made profound impacts on the HIV epidemic. They're incredibly robust and decrease the virus. So when you take your CART therapies as prescribed, you're able to suppress the virus completely in your plasma. And that's what I'm illustrating here. This is a graph that's showing you viral load in your blood, and it's looking at how much virus is there over time. So initially when you're infected, you have a high amount of virus, and current guidelines are to initiate CART therapy at diagnosis. This leads to a very rapid decline in the virus below limits of detection. And this makes the virus be able to be suppressed for a long period of time, up to decades. Now, the interesting thing about these CART therapies is that they're not curative and they require lifelong adherence. And if you stop these therapies for any reason, the virus can rebound. And that's what I'm showing you here at the end. And so while these CART therapies are fantastic, they are imperfect. And because of this, HIV remains a major problem today. And one of the things that we've become very interested in is the fact that HIV can affect, infect, and damage virtually every organ in your body. We spend a lot of time thinking about the brain, but many other organs also have damage during HIV. And the reason why we care about the brain so much is because it's a major target for the virus, and it's an organ that people tend to not think about when it comes to thinking about HIV. The brain is actually really important because it's infected within the first week of exposure to HIV. Some studies have shown you can find virus in the brain as early as 4 days after being exposed to the virus. And the brain is actually very unique. Compared to the rest of the body, the virus in the brain is driven by myeloid cells like macrophages and microglia, and to a lesser extent, astrocytes. The brain also represents a major viral reservoir wherein virus can persist in the brain even though it is suppressed in the rest of your body. So how does the brain actually become infected? Well, it's really interesting. What we believe occurs is you have some white blood cells called monocytes. These cells circulate in your blood, and their job is to go into tissues and to look for damage. If they see damage, they go into that tissue and try to begin to repair it. The problem is these cells can be infected by HIV. So if they're infected, they cross the blood-brain barrier, which separates your brain from the rest of your body, and they bring the virus with them. These cells can differentiate into another cell called a macrophage. And from there, they can release virus that can infect brain cells, like microglia and astrocytes. Not every cell is infected, but because they're exposed to the virus, they become activated and have a very strong immune response. These cells also produce a bunch of different neurotoxic factors and viral proteins. And so because of this, you have damage to neurons. And sometimes they can even lead to neuronal death. Depending upon the brain region where you have this neuronal damage, you have a consequent loss of function. And a very interesting thing is the fact that while these CART therapies are abundant in the blood, in the plasma, they actually can't traverse this blood-brain barrier very well. So the brain remains undertreated, even though the rest of the body has high amounts of CART therapies. Because of this, people with HIV develop numerous different kinds of neurologic diseases. In my group, we're really interested in thinking about cognition, and the term HAND, which stands for HIV-associated neurocognitive disorders, is an all-encompassing term that describes the spectrum of cognitive abnormalities you'll have when you have HIV. And it's characterized by some of these symptoms here. It can range from asymptomatic cognitive impairment or something that mirrors Alzheimer's in its most severe form. And the challenging thing about these cognitive impairments is the fact that CART therapies do not prevent it or treat it. So the incidence remains exactly the same, even when you have successful treatment. Now, certain groups of people are more at risk for having these cognitive impairments. One group in particular are those that use illicit substances. And this is actually really interesting because substance use is intertwined with the HIV epidemic. We've known for a very long time that it's a risk factor for acquiring the virus due to injection drug use and risky behaviors, but also it leads to worse HIV outcomes. People tend to have poorer control of the virus, and there's some immunomodulatory effects. So your immune system is a little bit different. Also, there have been many studies that have shown that substance use either accelerates or increases the incidence of various brain disorders when you have HIV. And this is because drugs of abuse seems to exacerbate all the mechanisms that I've discussed previously. However, the one thing that we really don't know much about is how drugs of abuse impact the ability of these CAR therapies from getting into your brain. And so we've become very interested in studying this and evaluating how are drugs of abuse impacting our efficacy in the brain. So I'll tell you two short stories that we've been looking at. So one of them is a translational study. We've collaborated with the Women's Interagency HIV Study, or the WISE study. It's a multi-site study throughout the country, and it is the largest study and the longest lasting study that evaluates the natural and treated history of HIV in women. These studies were done in collaboration with many people here at Hopkins, including Leah Rubin and Yan-Sun Zhu, as part of the Brain Health Program. And what we did was we evaluated about 3,500 women living with HIV, and we collected all their sociodemographic, behavioral, and clinical characteristics. And our computational collaborator, Yan-Sun Zhu, did trajectory clustering. So what she did was she made algorithms that allow for us to identify homogeneous subgroups of women based upon their demographics. So this allowed us to identify groups that had very comparable phenotypes. And once we had that, we wanted to evaluate different outcomes. We wanted to look to see if there were any differences in how these women had different brain disorders. We focused on women that had well-controlled virus and didn't really have any issues besides having HIV. They were otherwise very healthy. And our second group were women that used illicit substances and as a consequence had poorly controlled HIV. And we found something very interesting. We looked at cognition, among others, but I'm going to discuss cognition. What you see on the left-hand side are those that were well-controlled HIV, and on the right-hand side are those that use illicit substances and have poorly controlled virus. We're going to see a bunch of different circles. You see them grouped by color. The colors indicate the different CART class. So you see NRTIs, NNRTIs. These are two different kinds of reverse transcriptase inhibitors. And in the middle, you see the names of individual ART drugs. You also see on towards the middle of the screen some brown dots. These brown dots are going to indicate the different cognitive domains that were evaluated. So looking at memory or attention and working memory, executive function. And then you're also going to see some lines drawn between the ART drug and the cognitive domain. The weight of the line indicates the magnitude of the association. And then you're also going to see if the line is going to be red or blue. If it's blue, it's going to indicate a beneficial effect. And if it's red, it's going to indicate a detrimental effect. And what you see is very different patterns. If you have well-controlled virus, you see one, you mostly have ART impacting motor function, as opposed to if you're using illicit substances, your memory is impacted. So very different brain outcomes. You're also going to notice that different ART drugs have different effects on these two domains. One drug, for instance, seen on here, Maraviroc, is very strongly negatively associated with worsening memory. Whereas if you're well-controlled virus, it seems to improve motor function. So it goes to show that depending upon the group that you are and your different lifestyle factors, you're going to have a differential impact on brain function. And the ART drugs may help or adversely affect your neurologic function. And so with that, I'm actually going to stop. We're just about at time. I want to make sure that we have enough time for discussion. So thank you for your attention, and I'm really excited to answer any questions that you may have.
Okay, great. Thank you very much. So again, panelists can type their questions into the Q&A, and I'll just start. That last slide is really fascinating, right? So you can have a single drug within a broad class of inhibitors that can have a highly detrimental effect on one type of neurological function, while the others don't have anything, right? So, and they will have it in one population, not another, right? So you've also got this. So how do you think about putting this into some kind of practical screening use? I mean, obviously there's more data to collect. You have to, but I'm just curious to get your thoughts on that.
Certainly, this really speaks to the importance of personalized medicine in really trying to find the right drugs, right, for the right group of people. It goes to show that while these drugs all work fantastically at suppressing the virus, they seem to have different effects on brain function depending upon your different demographics. And so perhaps if you're at increased risk for having neurologic decline, we need to really think about which therapies we put you on. And we look at this among different groups of people as well, like those that were young versus older, or those that had comorbid hepatitis C, like, and all of them had different outcomes. And so I think we have to think about those comorbidities that people have, rather than thinking about them as something that makes the data kind of dirty, so to speak, making it challenging, right? We need to take that into effect and really see how that's going to intertwine with the therapies and other life factors in thinking about the brain.
Great, thank you. So there was a question that was posted, I think by Miranda Hitti, and it disappeared all of a sudden. So sorry, Miranda, I'm not sure what happened. If you want to post it, please post it back up. In the meantime, Peter Weiss writes, I'm sorry that I missed the earlier part of your talk. Can you provide or revisit the numbers of people affected by these types of neurological effects of HIV?
Fantastic question. So the prevalence is about half of people that have HIV have some spectrum of neurocognitive deficit, but that is a range, right? It can be asymptomatic, which means that you have some cognitive abnormality, but it doesn't impact your daily living. It's actually only found out upon diagnosing and getting neuropsychological testing. The next severe is mild cognitive impairment that doesn't interfere with daily living. And then the most severe is HIV-associated dementia, which is likened to an Alzheimer's type dementia, but that's very rare now because of these CAR therapies. There's a spectrum, but about half of people will develop some kind of abnormality, which means it's a really big problem.
Right. Great, thank you. Okay, so Miranda's question's back up. Does this happen with alcohol and prescription drug abuse as well?
That's a really fantastic question. Alcohol, we haven't really been able to parse out, and it's just because it's so widely used. It's really hard to get people that don't drink anything and compare them to those that do. But I'm sure it does, because alcohol has similar effects. Prescription drugs do have another paradigm that's very comparable to this. We're beginning to look at those. And I think that's really interesting because we think about illicit substances, but those prescribed drugs do in fact impact many systems in your body and can have effects on brain function. So we're beginning to look at those as well in a very similar fashion.
Okay, great, thank you. All right, so one last question. Why, this is from Alexandra Benestek, why do some individuals not experience neurological issues? Does this just have to do with the blood-brain barrier?
Yeah, that's the question that we really want to understand, because as of now, we don't have any anything in clinical use to predict who's going to have cognitive impairment or not. Some hypotheses are things like your cognitive reserve, so just your own intrinsic factors, right? Your own background and genetics. Some things like that are social demographic, like education, but also things like background and trauma, those kinds of things impact your brain certainly and can make you more predisposed or not. But really, at this point, we're just making predictions. There's nothing at this point that we can use to identify who's at risk. And it's a major factor that we need to figure out.
Okay. All right. So last question just came in. So Barbara Jungwirth, some experts question the importance of asymptomatic hand. Could such a diagnosis exacerbate stigma without impacting treatment?
Really, really good point. So there's been a lot of effort to really figure out what this asymptomatic hand is. And it actually, even though the name is asymptomatic, it does impact you. The challenge with this being able to identify your quality of life means you have to have the ability to say, yes, I think I'm more forgetful than I was before. So it's your own self ability, and that's subject to bias. But also, even if you are asymptomatic, it's been shown that diagnosis leads to poor HIV outcomes, less likely to take your CART therapies, lower lifespan. So even though it's quote unquote asymptomatic and not that profound impact on your daily life, it is bad. And it also seems to be making you at increased risk to go to those more severe forms of impairment in time.
Wonderful. All right. Well, thank you again, Dionna.